Hi guys, today we're going to look at genetic modification in potatoes that is going to make them resistant against late blight or potato blight. And potato blight was the cause of the Great Irish Famine and other famines that destroyed a lot of potato crops.
So, first of all, we looked at many studies referring to the late blight or how to confer a resistance to late blight in modern potato events. So, we looked up a very important and very famous late blight resistance protein, which is um, prevalent in several potato or solanum plants in the same genus as our commercial potato. And we looked up at UniProt the protein sequence of the late blight resistance protein, and then we back translated the amino acid sequence with reverse translation tool, which we will link in the description down below. And we optimized the sequence, or like the codon, or we optimized the codon sequences for a solanum or like for our potato event to ensure a effective translation in potatoes. So I will look up it for a long time to actually find it.
Yeah, and one advantage of using something so closely related to the commonly used potato is that it's basically seen as a cisgenetic event, and the probability of that being admitted to a market is way higher. For example, in the Netherlands,
Right, to be approved.
yeah,
To
yeah, cisgenic events sometimes get approved at least, right?
It's more easy to let a cisgenic event to be approved in the EU than a transgenic event, because using something or like a gene of a closely related plant is more easy to be approved in the EU and even in the US. So we were more specific to find something in a related plant.
Yeah, and
so here we start to insert the sequence which we just got from the back translation, reverse translation, and added the stop codon sequence, which is not translated with a protein, of course, into Snapgene in the viewer, and then we go further. And of course, even if it's very long, we still show it in a linearized way and not in a circularized way, because that would be like a pseudo plasmid. So here we add a feature just for the reason that we can then track the sequence, and we enter the name of the protein sequence which would be expressed, and then the name of the plant which from which it comes.
yeah, and the feature is basically just like an annotation in Benchling, because the program you see here is Snapgene.
Right, then we translate it in sequence view so we can see the protein sequence and if it's even get expressed. And then we start the gateway cloning. So we need to insert on or near the BP gates to then insert it or clone it into our donor vector. Right, we first need to select the whole protein sequence, need to look up if it's still in line with the stop codon. Right, it's completely in line. So we go to the donor vector, select our donor vector. In this case, P donor 221, 221. We can see that the donor vector has like ATTP gates to be cloned and a lethal factor called CCDB.
Yeah, yeah, and yeah.
So now we choose our primers. Right, now we can look up how the cloning into our our donor plasmid gonna be performed. the default selection will be around 60 degrees for the primers to take place. And we will stick to the default settings because it's very good for like PCR primer annotations or primer events. So and then it shows us the whole history, or like shows us how it would work, and even sends a different file with a primer sequence. So we can see it's now in our vector. We can see the history tab to see what actually happened. We can see the annealing primers to then insert the gates, our B gates, that then are incorporated with our P donor sequence with the P gates to fuse them to get like L gates that we need to use for our destination vector cloning event.
Yeah, which this isn't yet.
So and now that we have our donor vector and our gene of interest inside of the donor vector, we now can perform the LR cloning of one fragment. And so now we can choose our destination vector, and that in this case we will choose P early gate 100. It works best for our case right now. It also has a lethality factor, which is then replaced with our gene of interest. Now we can express the cloning event. And we finish up the cloning from our donor vector and our destination vector. We can now see we have even the cloning which we performed earlier from our earlier event to get it into our in our donor sequence or in our donor vector, and then the event that took place, the LR cloning, the gateway LR cloning from our donor vector inside of our destination vector. And it's expressed, and the tDNA repeats. It's completely in line with the tDNA, which will be incorporated by the agrobacteria transfection event inside of a random place inside of the plant genome. We are using the cauliflower mosaic virus 35S promoter sequence and terminator sequence. We even gonna use, we also gonna clone inside of the plant genome bleomycin resistance selective marker.
Okay, so this might still have been a little bit confusing. So we're going to take a look at this infographic. So what you're going to do in this protocol is anneal to one ATTP1 and an ATTP2 sites to the sites of your gene of interest, and then you're going to choose a donor vector that contains the ATTP1 and ATTP2 sites and CCDB in the middle, and the CCDB is a lethality factor, and if that's expressed in bacteria, they instantly die. So if this plasmid gets into bacteria, they will die. But if the BP reaction of the gateway cloning using BP clonase works right, then the CCDB will be excised and only the gene of interest will be contained, flanked by ATTL1 and ATTL2. That's just like how gateway cloning works. So ATTP plus ATTP result in ATTL. And then we also chose afterwards to do the LR reaction, where a plasmid, an entry clone containing these LTTL sites flanking the gene of interest, gets combined with a plasmid containing two ATTR sites, again flanking a CCDB lethality factor, and but this is another plasmid. So, but it's somewhat similar. And if you add those two together with the LR clonase, you get two products. One is the expression clone, which contains everything that you want, and it will also in this one the gene of interest will be flanked by ATTB1 and ATTB2 and a toxic byproduct, but that doesn't really matter because if the toxic byproduct gets into bacterial cells, they will instantly die. And so you will get bacterial colonies that will only contain the plasmid you wanted to.